Good morning, good afternoon, good evening, as always, for the GVN. And welcome to this Forefront of Virology webinar. And before introducing Dr. Ulrike Protzer, as always, I just want to say a very few words regarding Global Virus Network because some of the attendees are not necessarily aware of it. So briefly, this is a science-driven and independent network which has been created in 2011 by Bob Gallo together with B.D. Hall and Reinhard Kurt. And three main activities: research with what we call the task force and the watch groups. You have all the details on our website. Second, education and training with the GVN Academy. We have the capacity now to support young investigators with fellowships and mentoring activities. And third, communication advocacy. And it's really in this context, and also a lot of partnership with industry, which is really very important for the GVN. So it is really in this context that we have this series of webinars. And also in this context, it's a great pleasure to welcome Ulrike Protzer. Ulrike is the director of the Institute of Virology at Helmholtz, Munich, and the Technical University of Munich, and she holds the chair of virology. She has been trained in Germany, in Switzerland, and South Africa. And really, Ulrike has always been at the interface between infectious disease, hepatology, virology. And her laboratory has always merged molecular virology and immunology to really get a sense of interaction between the virus and the host. Her activities have been very much focused on hepatitis B. She has provided many very important contributions, and she is really an internationally highly recognized expert, in particular on immunotherapy and vaccines. But she has also been working on COVID-19, and actually she gave a very interesting lecture, short lecture during a COVID-19 task force of the Global Virus Network. And again, she has produced very interesting results. Finally, Ulrike is very much dedicated, I would say, to coordination and action for the best of research. She has been the founder of the German Infection Research Center, and she has always been extremely active for the Global Virus Network, and actually is the head of one of the European centers of the GVN. So with this, she will discuss today one of maybe the major topic regarding HBV. Can we hope or not for an HBV cure? And again, it's a great pleasure, Ulrike, to welcome you for this webinar. And thank you so much for giving the lecture.
Yeah, thank you very much, Christian, for this really nice introduction. I would share my screen if that's okay. And I hope you can see and hear me fine.
Yes, we do.
Super. So thanks a lot for the opportunity to discuss with you today, HBV cure, is it fact or is it fiction? And maybe give you some updates or insights into where we stand with hepatitis B virus research and also cure at the moment. Basically, there is two different fates of an infection. The first one is an acute, often self-limiting infection. That's when you acquire the infection as an adult. The other one is a persistent, often lifelong infection, and that's very often the case when you acquire the infection as a young child, either by vertical transmission from your mother or as a toddler. And of course, this year is the big problem we do have, because that's also very hard to interfere with prophylactic vaccination efforts, which are certainly extremely important, but will probably not be able to completely eliminate the virus, but we can discuss that later. So although we have a prophylactic vaccine, almost 4% of the world population still carry hepatitis B virus. And that's a major health threat, obviously, because 880,000 people die of its consequences. And I showed you the numbers in the different countries. And when you look at this, then you see that in America, it's relatively few people. And that's probably why for a long time, it was sort of not the focus of research. And I always say it's the most frequent neglected disease of the world. Why? If you look at how malaria, how HIV, and how tuberculosis develops, then you see HIV's with its fatalities, so that's numbers of deaths, which are shown here in this work by David Thomas in the New England Journal, is on the way down. Malaria is decreasing, tuberculosis is increasing, but viral hepatitis is still rising. And if we do not do anything, it will largely outnumber the other causes of infection-mediated death. And that's why the WHO has recognized that problem and really is taking action here. Why is that the case? Why does hepatitis B virus kill so many people? Well, very often it causes mild symptoms for a long time, 30 years, sometimes 40 years. And then patients, if there is inflammation, can develop liver cirrhosis. But even in the absence of liver cirrhosis, as you can see here on the right-hand side, hepatitis B virus can cause hepatocellular carcinoma. And that's why it's still the #1 death or cause of death from hepatocellular carcinoma. And that's the big problem we do have. Now we have antivirals available. These are mainly nucleoside analogs. They act at the step of reverse transcription, which in the hepatitis B virus life cycle is relatively late. So the virus attaches to its receptor on a permissive cell, is uncoated. The capsid transports its content, the viral DNA, into the nucleus, and in the nucleus, this partially double-stranded DNA is filled up to a so-called cccDNA. And that's an episome which normally is very stable in the nucleus of an infected hepatocyte and can remain there for up to 5 years. That's the transcription template. And from there, RNAs are transcribed. The longest one is encapsulated and reverse transcribed into new viral genomes, and then can either be re-imported to fill up the cccDNA pool or be exported as variants. Now, nucleoside analogs block that process of reverse transcription, and thus they block the production of new variants. Unlike in HIV, where they act at an early step in the virus life cycle because reverse transcription occurs upon virus entry, here it acts at the late step of the virus life cycle. And to understand that is important to understand why it's so hard to achieve a cure. So nucleoside analogs and also capsid assembly inhibitors, which are under development at the moment by a number of companies, act here at that late step and have very little chance to cure the virus, so to deplete the cccDNA persistence form. Then there is interference. There is TLR agonists, which activate basically interference and other cytokines, and there's siRNAs. Those act a little earlier because they already act at the level of the viral RNA, but still leave the cccDNA untouched, more or less. Interference in a small portion of treated patients can cure the infection. Newer studies say between 2 and 5%, but they have relatively high side effects. So doctors do not like to give that anymore. Then we have entry blockers. They prevent the infection of new hepatocytes. But again, cccDNA persistence is not affected. And so far, clinical trials did also not show an effect on hepatitis B virus, but they did show an effect on hepatitis Delta virus though. And the last, not least, is trying to lower the viral antigen production, which is S and also E antigen. And that can be done again by siRNAs or by so-called nucleic acid polymers, which block the secretion of S antigen. Now, as you may appreciate, these directly acting antivirals control but do not eliminate hepatitis B virus. And that's what clinical trials show so far, a significant hepatocellular carcinoma risk still remains. So the cancer risk remains high. And that's why cccDNA targeting would mean targeting the core issue of HPV cure. And when you think about induction of hepatocellular carcinoma, it's not only the cccDNA persistence despite antiviral therapy, which causes an ongoing inflammation in the liver, but it's also that due to a mistake in the virus life cycle, linear genomes can be formed, and these are prone to integrate into the host genome. And the cells which carry these integrates, they are thought to be the epicenter of replicating hepatocytes and also carcinoma development. Now, what can we do? How can we then cure HPV if the direct antivirals have a hard time to do so? Well, basically, you can kill infected hepatocytes. You can also try to induce cell division, because the cccDNA, unlike the herpes virus genome, does not have an active nuclear retargeting. It always has to go via the capsid. And you can try to induce a degradation of the nuclear cccDNA. And all of that would be activated or induced and done by immune therapies. And that's why we usually focus, or we believe that some sort of an immune therapy will be essential to really achieve an HPV cure. Now, when you look at self-limiting infections and immune control of HPV, it has been described by Luca Guidotti and Frank Cisari that part of the control is done by cytokines. This is old work where there was an adaptive transfer of T cells into HPV transgenic mice. And you may appreciate that at day 5 in these mice, HPV RNAs, but mainly also the replicative DNA forms in the liver are completely diminished. But when they block cytokines like interferon and TNF, this can be restored. In the transgenic mice, there is no cure possible because you would have to kill the whole mice to cure the disease. But it shows the role of cytokines, and it shows that there is a non-cytolytic control of viral infections. And they also show that that's by innate and adaptive immune responses. And this, if you look now into what a self-limiting infection in humans look like, is correlated always by neutralizing anti-S antibodies. So the patients seroconvert from S antigen as a sign of virus persistence production to anti-S neutralizing antibodies. Patients who have cured an infection, they have an activation of T cell immunity. And this is usually accompanied by a limited ALT flare and a limited hepatocyte death. That means a limited cytolytic immune control. And that's probably why gene expression from the remaining cccDNA and or HPV integrates is suppressed, in addition to the cytolytic killing by the non-cytolytic control induced by cytokines. And we wondered, how does that work? And we found a while ago that the cell has an intrinsic ability to target a nuclear DNA, and in our case, the cccDNA, and that works by activating deaminases. So basically, if that's the viral cccDNA persisting in the hepatocyte, and you activate deaminases, for example, by interferon type 1, or by some, in our case, we use the lymphotoxin beta receptor agonist, which activates NF-kappa B dependent pathways. You activate different types of deaminases called ApoBec C deaminases. And there is one which is called ApoBec 3A, and this ApoBec 3A is induced by interferon, and it's an interferon-induced gene, and that's essential to degrade cccDNA. And we've recently shown that a second, also ISG, so interferon-stimulated gene, the ISG20, is necessary then to not only modify, but finally degrade the cccDNA. And if you activate the NF-kappa B pathway in cells, then it's another deaminase, it's the ApoBec 3B deaminase, which initiates finally the same process. Now, what do we think a curative treatment for hepatitis B virus could look like if antivirals don't do it? So what I personally think is that we need nukes or maybe alternative antivirals for at least 6 months to reduce the number of infected cells to its minimum. And then we need some sort of an immune trigger. And this immune trigger then, again, after a while, will be able to achieve a functional cure or an immune control, or ideally a complete cure. The big question is, would the cytotoxicity and the liver damage that's probably associated with that be acceptable? Now, we tested different options for potential immune therapies. We started off with cytokines. However, the problem with cytokines, if you use them for treatment, is that the dose which is tolerated is always limited, and that accounts for all of them. We also tested PRR agonists, TLR agonists, RIG-I agonists, but the same thing. As soon as you pile up your dosing, you start to get side effects and you're limited. And it's very hard to reach a curative level of cytokines in the liver. Checkpoint inhibitors, others tested, and there's also limited success so far. But what we think is, or believe is much more promising, is really having an antigen specific, that means targeted approach, because then you activate ideally T cell or HPV specific T cell immunity, and that does both. It secretes cytokines at the site of interest in the liver where the infected cells are, and of course it kills infected cells. And when you want to do so activating antigen specific targeted immune therapies or immunity, then you can choose 3 ways: doing that in an antibody mediated fashion, use T cell redirection, or design a therapeutic vaccine. And I'll guide you through about those 3 options, because basically we tried all of them. Let's start with T cell therapies. So function of T cells, effector function of T cells is very obvious. They would kill infected cells and can also kill, ideally, HPV-dependent carcinomas, but they also secrete cytokines and would have an effect on the virus by cytokines. And that's the publications which show the 2, or decipher the 2 effects, the cytolytic and the non-cytolytic effect, which we have here by the T cells. So HPV-specific T cells can fight the persistence of cccDNA simply by killing infected hepatocytes, and also by local cytokine production, which purges the cccDNA in the patients. And then HPV-specific T cells may also find hepatocellular carcinoma. That means they can kill HCC cells, which carry the HPV genome as an integrate, and the cytokines can alter the microenvironment and help other T cells to integrate. When you think about how can I do a T cell therapy, then you end up with either TCRT cells, or recombinant T cell receptors, which you put onto a T cell, or CAR T cells, so a chimeric antigen receptor, where usually a single chain antibody or an antibody fragment is the recognition domain. Now, TCRT cells would obviously recognize their target peptide on an MHC, so that's a processed antigen. And whereas a CAR recognizes a native antigen on the surface of infected cells. Now, luckily, HPV has the surface antigen, and this is also displayed on the surface of infected cells, as I'll show you. So we could design as a CAR against S, and we could clone T cell receptors. And what we could also do once we had the single chain fragments active here, we could, instead of using those to generate a CAR, so a chimeric antigen receptor, we could use that to design T-cell engager antibodies. Now, coming to the target, why is the viral S antigen a good target? Because first of all, it's found on the surface of cells. In this case here, it's infected hepatocytes, but you also find it on the surface of tumor cells, as you can see on this sections from patients which have developed HCC during their chronic infection. Second of all, it's always the last antigen which is expressed, or one of the two last antigens. So it's still expressed once the patient has developed a cancer. That's why we chose it. And we showed by immune or fluorescence microscopy studies that you really find this as antigen stained here with different types of antibodies, a monoclonal antibody, and a polyclonal antibody on the surface of cell, that this overlaps with WGA as a surface marker. So you clearly find as a target on the surface of infected hepatocytes. And then we said, okay, we need a single chain antibody. And we cloned or fished one out of a B-cell library of vaccinees. And this single chain fragment we call C8. And to test that, we put that onto the constant domain of an IgG antibody to have something which we can test. And this was enabled us to define the epitope, which is a conformational epitope here marked in blue. And we used that to show that this single chain fragment would bind all the different genotypes which circulate. And then we did electron microscopy and used this MOMAP, how we called it, to stain. And we found S antigen on the surface of HPV producing hepatoma cells. This is an example of the HepAD38 cells. This is HepG2-15 cells. And we also found it on the surface of cells infected with HPV, as you can see here, but not on HPV negative controlled cells. So that seems our binder, which we had identified, seemed to be a suitable binder, and our target seemed to be where we expected it. So what we did is we generated a chimeric antigen receptor using this single chain fragment as a binder, and in the first place used a second generation CAR design. And the advantage of such a CAR would be that obviously you can treat patients independent of the MHC type. In our case, because we have a binder that recognizes all HPV genotypes, also independent of the strain which infects the patient, you would not expect any TCR mispairing and be independent of antigen production. But there would be a risk that circulating soluble antigen, which is there as antigen, you could theoretically overactivate the T cells, and that's something we needed to test. So what we did in the very first experiments, we worked together with surgeons who had access to liver samples they had to excise due to metastases of colorectal cancer. And we isolated hepatocytes, primary hepatocytes from these liver sections, which we got. And from the very same patients, we got PBMC isolated T cells and grafted with our CAR constructs the T cells and infected the hepatocytes with HPV. And what we found is that infected hepatocytes, but not uninfected control cells, activated T cells to secrete interferon gamma and IL-2 when we put an S CAR on the T cells, also when we put an L CAR on the T cells, but not with control CARs or non-transduced CARs. And the most important thing, we achieved an CCC DNA cure in our hepatocyte cultures when we used our S CAR constructs. And this approach then was very promising, and we went on and brought that into in vivo into animals, and we showed that the SCAR T cell treated animals, they continuously dropped in their antigen levels, whereas control or non-signaling CAR treated animals, they didn't do anything. Now then we wanted to know, okay, would that approach now also help? Obviously, it killed the infected cells, so there was a cytolytic effect, but would that also help to activate this non-cytolytic effect, which we had described? And just to remind you, we have either interferon type 1 or type 2, which then activates ApoBec3A as a deaminase. That goes to the CCC DNA, deaminates the CCC DNA. The uracils which come up are prone to excision by specific enzymes causing a DNA damage. And then this DNA damage, or this damaged CCC DNA, can be recognized by a second interferon-induced gene, which is ISG20, which serves as an exonuclease and degrades the CCC DNA. So we've published this pathway and wanted to know, would that also be active if we now have our CAR-grafted T cells? And to test that, we used infected hepatology cells, and we treated the infected hepatology cells with interferon gamma and TNF mixture. And we also cultivated them with the CAR T cells. Now, what happens, obviously, is in the co-culture, you have a cytolytic effect. So the target cells are killed. If you just have cytokines, there is nothing which kills, so you would have a non-cytolytic effect. And then we asked the question, if we now physically separate the CAR T cells from the target cells by a transval, would then we also get a non-cytolytic effect of the T cell cytokines secreted if we stimulate the T cells in the transval? And what happens if we do not stimulate them? And the result was very clear. So if we look for either CCC DNA directly or E antigen as a marker of the CCC DNA infection, then we find, compared to mock treated, in the co-cultures where there is a cytolytic effect, a drop in CCC DNA and E antigen. That's what you would expect. But we also found that when we only put cytokines, but not T cells, onto the cells, and we found it if we separated the T cells from the infected cells by a transval and stimulated the T cells on the transval, but not if we did not stimulate them. So very clearly, there is a cytolytic killing by the CAR T cells, but also a non-cytolytic effect by the CAR T cells. And this was later on then observed in, or confirmed in, publications by other groups, Antonio Bertoletti's group, for example, and Maura Dandry's group, which showed that the TCRT cells contribute to cytolytic and non-cytolytic elimination of HPV in humanized mouse models. So we thought, okay, that looks quite nice. Maybe we can make that approach even easier so that we do not have to handle T cells in a GMP environment and put a CAR on it. Maybe we can, instead of using a CAR, which is brought onto or expressed on the T cell, we can simply use a T cell engager antibodies. So antibodies which would bind our target on the infected cells and would attract T cells by CD3 and CD28 binding. And we did several different constructs, but the main constructs are shown here, something which we call bimaps. So they carry our binder to S antigen, and on the other end of the antibody constant domain, we've used either a single chain recognizing CD3 or something recognizing CD28. And we call that a CD3 or a CD28 bispecific antibody or bimap. And what we found is if we combine those bimaps, then we can activate T cells to secrete different cytokines. And we can activate T cells, and in this case, it's non-transduced T cells. It's simply T cells from a healthy volunteer. We can activate them to kill HPV positive target cells. And this is in a dose-dependent fashion. So the more antibody we titrate in, the better the effect is, but you don't need too much. And it's also that we can convert CD4 as well as CD8 T cells, as shown here, into killers using that T cell engager antibodies. And that works quite efficiently. And we just last year published that it's as efficient as using CAR-grafted T cells, which is very nice. Now, the question obviously was, can we now eliminate HPV with this approach? And we went again into cell culture, infected hepatocytes with HPV, and then incubated PBMC of a healthy donor onto our hepatocytes and added our antibodies in different doses. And adding in the antibody in different doses clearly showed a dose-dependent effect of T cell activation and also killing. And even when we went into in vivo and we used mice, in that case immunodeficient mice, into which we transplanted the target hepatocytes into one flank and HPV negative control hepatoma cells into the other flank, and then transferred PBMC, human PBMCs into the mice and injected our antibody combination, then we found that in those cells, which are target cells, so which carry the S shown in the lower panel, the BiMAP injection led to a significant reduction in quite a few mice, even to elimination of the tumors, whereas the control cells, which do not carry the target in the very same animal, they were not influenced in their growth. So that seems to be an interesting approach also to bring into the clinics. Now back to TCRT cells. So T cells which carry a cloned T cell receptor. These were first described by Adam Gehring from Antonio Bertoletti's lab, and then Karin Viskirchen from our group set out and also cloned a series of T cell receptors and showed that these work. Now, how did we do that? We used the PBMC from HLA-A2 positive donors with an acute resolved HPV infection, stimulated them with different peptides where we wanted to isolate the T cell receptors for, and then could identify populations growing out of specific T cells, sorted them out, identified their T cell receptor chain sequences, put those sequences into a retroviral vector, and expressed an artificial T cell receptor. And that's just to show you that here on this upper right corner, you can express such a T cell receptor on, again, healthy donor PBMCs. And then we compared all the different T cell receptors she had identified and found out there is two very efficient ones. One's against core, HPV core, and one against HPV-S, which we call 4G. And that's, for us, the most interesting one because we really want to target the S-positive cells in the patients, but also hepatocellular carcinoma based on HPV. So Karin first set out and tested those two T cell receptor T cells. And what she did is she infected hepatoma cells in culture and put her TCRT cells on and asked the question, do those TCRT cells cure the infection? And she went into mice. That's the results from which we found in cell culture. That's mock T cell treated cells. And then when you add different doses of the S20 specific TCRT cells on, or the core 18 specific TCRT cells on, you may appreciate that's the killing curve of the hepatoma cells. They are very efficiently killed, even at a dose effector to target ratio of 1 to 85, which is very low. It takes a little bit, but then the T cells take over and kill the infected hepatocytes. And that's not only CD8 T cells, it's also CD4 T cells, which do the job here. Now, the big question was, is there an antiviral effect? And yes, there is. Otherwise, I would probably not present the data. So e-antigen as well as S antigen secretion is completely lost, or at least below the detection limit in the treated hepatocytes. And also when we check CCC DNA levels, then that's mock treated, that's untreated, and that's the different TCRT cell treated CCC DNA amounts in the cell cultures, they're gone. And also rcDNA, which is a little better protected by the capsid, is significantly lower. But the big question was, would that now work in mice? And to show that, we teamed up with Maura Dandry's group in Hamburg, which have liver humanized mice. And what they did is they repopulated for us their mice with HLA-A2 positive primary hepatocytes. Remember, if you use TCRT cells, you are HLA restricted. With the CAR, you are not. So that's sort of a disadvantage here. However, the TCRT cells are a little more effective. So that's the advantage. So after repopulation, they infected the mice with HPV, waited a little bit, and then we sent over our TCRT cells. And in that case, we used a mixture of the S and core specific TCRT cells, and we analyzed the animals. And what happened is that looking into the liver and staining for HPV core here in green, shown in green, and those liver humanized mice have human hepatocytes shown in red, and mouse hepatocytes, which is the remaining, which is not red. You may appreciate that in the treated animals, you do not find a green, means not a core positive hepatocyte anymore. And also when we checked by RNA scope for HPV RNA, in the treated animals, there was hardly any positive hepatocyte detected anymore. They checked a lot of stainings. So that seemed to be very efficient and was the basis then for us to say, okay, that's really interesting receptors and we should bring them into the clinics. So the idea here is that if you have a patient infected with hepatitis B virus, and you would have a patient which has an acute hepatitis B and can clear the infection, they would have a lot of T cells, so an effective T cell response. Whereas in the chronic hepatitis B patients, you have very weak T cell response and T cells are really scarce. Now, what we would do is we would isolate T cells in that situation, ideally before the patients develop their hepatocellular carcinoma, but latest then. We would graft the T cells with the right T cell receptors. We would expand them and we would reinfuse them back into the patient. So doing a classical adoptive T cell therapy. And to be able to do that, we founded a company which is called SCG Cell Therapy. It sits in Singapore and works a lot also in the Asian room. And we teamed up with the different Chinese investigators, and we were able to treat the first patients with this approach. And that's the results which were presented last year at the EASL meeting, so the European Association for the Study of the Liver Disease. And we had 3 patients, HLA-A201 positive, which received our TCRT cells, either 5 × 10⁷ or 1 × 10⁸ per kilogram. And what you see is that the S antigen level in those patients treated in 2 of the 3 rapidly dropped and remained negative. That's the second patient, rapid drop remaining negative. In the third patient, though, it was not that effective. We had a drop, but then S antigen came back and there was only a minor effect. In all 3 patients, there was obviously, that were patients with hepatocellular carcinoma, and the goal was to treat those finite stage patients which had no other therapy option anymore. And the cell infusion obviously caused some sort of hepatocyte damage. That's the ALT level, but this was transient in all 3 cases which we observed, and correlated very well to the reduction of S antigen. So 2 of 3 patients eliminated the S antigen, and this was even more encouraging, we found a stable disease or even a slight reduction in the tumor size in these 2 patients which lost the S antigen, whereas the 1 patient which did not respond also did not show an anti-tumor response. And this is now the basis for a systematic phase 1 trial, which has been approved in China and in the US, and enrollment of patients has started. So more about that maybe in a year or 1.5. But to sum up here, I would like to show you just a few more slides on a third approach we are following, and that's a therapeutic vaccine. And what the idea of this therapeutic vaccine is that what you have in self-limiting infection, an immune control, meaning strong antibody and effector T cell responses, is absent in the immune tolerant situation of a chronic carrier. So there's new neutralizing antibodies and T cells are really scarce. So what we want to do is to turn this stage into this stage. So an immune tolerant into an immune control stage. And that's why we use the therapeutic vaccination to activate T cells. Now, the big question is, does that work? And the answer is, we don't know yet. There have been a lot of trials done. This is one we are pursuing here, but none of them has been very successful so far, although the first press releases are there that there might be effects. However, they were not ideally defined. Why? Because you have a lot of different means for vaccine components to be used. And what we decided on for our trial is to focus on a combination of proteins, but not single proteins, but VLPs, and a recombinant viral vector. And in our case, we decided for an MVA vector because that's very good in boosting T cell responses. Now, we used those and first tested them singly, the MVA singly, the protein singly. But with the MVA, we only got a pox response, so no monkeypox in these mice anymore, but almost no HPV response, and only protein gave very little response. But when we now combine the two in a heterologous approach, prime boost, using protein prime and MVA boost, then we got very nice responses, T cell responses, as well as antibody responses. So we thought, okay, that's something which we should bring into the clinics. And we are at the stage of doing that. So we want to go into nucleoside analog pre-treated patients and then twice vaccinate them with the particulate protein, S, and core antigen in a particulate form to activate B cells and to activate CD4 T cells. So that means ideally the patients would now develop neutralizing antibodies that prevent the further spread of the virus, and we would prime T cell responses. And these T cell responses can then be boosted by the MVA vector, which is designed to be applicable all over the world, and that the CD8 T cells do the rest. So they kill infected hepatocytes and secrete cytokines and do what I've explained to you. And this is just one example that works. We have generated a mouse model where we, with AAV, bring HPV into the liver of the mice, and we have wild-type HPV and E- HPV. And these animals, as expected, the wild-type HPV infected animals secrete E antigen, the other ones didn't, but both have S antigen. When we observe over time, that's a very persistent infection shown here for 3 months, but you can look for even more. Now, when we vaccinate those mice, what happens is they seroconvert to anti-S. They lose their S antigen, irrespective of E plus or E minus status. The E plus mice, they lose their E antigen, and both groups of mice have a transient elevation of ALT, showing that while the infection is cured, there's only a limited inflammatory response in the liver. And now I think due to time reasons, I'll skip the next 2 slides, but just show you this slide. We also thought about which is now the important T cell population doing that. Is it CD4 or is it CD8 T cells? And if we deplete CD8 T cells, then we still have an effect of the vaccine. So that's the isotype control where we see a loss of S antigen, a loss of E antigen. But if we block CD4 T cells, then the effect of the vaccine is blocked. And this has shown for the first time that it's really the activation of CD4 T cells, which is very, very important for the success of a therapeutic vaccine. If you're interested, the manuscript has just came out in Journal of Hepatology, accompanied by a second paper here. So the activation of CD4 T cells during prime is really essential. And that's also why our approach designed to primarily activate CD4 T cells, I think, is very promising. Now, the last thing is what we found is that the level of HPV in the liver and as antigen in the blood is very important for the success of a vaccine. So we can titrate in HPV into our mouse models and either generate low replicator, medium, or high replicator mice. And if you vaccinate those mice, in high replicators, there is not a big effect. In medium replicators, you see some effect, and the low replicators, they cure. And this is inversely correlated with the T cell response. So the high replicators have almost no T cell response, whereas the low replicators have a very nice T cell response upon vaccination. So the high HPV antigen levels impair the efficacy of therapeutic vaccination. Now we thought, okay, there is something you can treat high antigen levels with, and that's siRNAs. So we teamed up with Alnylam, and we used siRNAs against HPV to lower the antigen levels of S and to lower the antigen levels of E. But if we stop treating, all those mice had relapses and basically had the same level of antigen expression as they had on the beginning. So siRNA per se, apparently in our mouse model, but also it seems as now in patients, does not do the trick. But what if we now combine with our therapeutic vaccine, because now we were able to convert high expressor mice into low expressor mice. So we did that and we vaccinated. And what happened is the mice where we pre-treated with siRNA and then vaccinated them, they cured. And they persistently cured. They developed T cell responses as expected. The others did not. And they cured HPV. And this you can see best, I think, here in immunohistochemistry stainings, where only as siRNA therapy, there is a lot of HPV infected cells left. And that's why all these animals have a relapse and end up back here. But the combination with therapeutic vaccination, you do not have any infected hepatocytes anymore. And that's why they're cured. So the outlook here is that we also want to test this in the clinics, obviously. And we want to use our therapeutic vaccine that activates B cells, CD4 and CD8 T cells, because we think that a targeted design is really important. In our case, that's the heterologous prime boost. The design we chose covers HPV genotypes A to E, so can theoretically be applied worldwide. And that's where we want to start that. So in phase 1A and then 1B to A trials. And we hope that the phase 1A trial can start in about 3 weeks. That's at least the idea. We still have the final granting of the trial protocol pending, but we could address all the requests from the regulatory agencies. So we hope it's really going to start very soon. However, in initial trials, we may have to pre-select low HBs antigen patients until we're able to maybe then combine with siRNA and follow-up studies once we've shown. So in summary, I've shown you that HPV is able to avoid and escape immune responses, that we have antivirals that control but do not eliminate the virus, that immune activation, at least that's what we are convinced of, will be necessary to clear the virus, and that therapeutic vaccination activating virus-specific T cells seems most promising. Why? Because it's the easiest. You do not have to handle T cells in a GMP lab, and it's also the most easily and broadly applicable approach. T cells are very scarce in chronic HPV infection, so it seems as if new T cells are required, either by adoptive transfer or by therapeutic vaccination. In our mouse models, therapeutic vaccination is safe and can cure HPV. Obviously, we have to prove that in humans. The activation of CD4 T cells is important to help B and also CD8 T cells. And if you want to go for established HCC, which is based on hepatitis B virus, then probably therapeutic vaccination will not do the trick anymore. But there, you really would have to go for T cell therapy, where you can directly infuse the pre-activated and specific T cells. And we've shown that it can cure HPV and restrict HCC growth, whether it can also be curative for HCC. That's what trials have to find out. With that, I'm at the end, and I thank you very much for your attention, and I think everybody who contributed to these large set of experiments, so the people who did all the work in the preclinical mouse models, the people who did all the T-cell identification and work, the people who helped us with the mouse experiments, the project development team who does all the regulatory effects, the immune monitoring, which is important, our clinical partners who help us in developing, our partners at SCG Cell Therapy, and of course, at the end, most importantly, the patients who are willing to participate in clinical trials and help us to find out whether that's really efficacious. And that's all a team effect, a team effort. And that's why all the credits go to the teams here, which did the work. Thank you.
Thank you so much, Ulrike. Great presentations and many very stimulating results. So this paper is open for discussion as always, and you can post some questions on the Q&A if you have. And well, maybe just to start, Ulrike, I mean, just to be clear, when you discuss S, you are always really discussing S without pre-S.
Yes and no. In our vector-based vaccine, there is also pre-S to activate T cells against pre-S as well. When I say S as a marker, as a diagnostic marker, it's always the S antigen, which is secreted and that mainly contains the small s. When I talk about S antigen as a protein prime, then it's only the S, because it's the just very conventional yeast-derived S antigen.
Okay. And yeah, no, I understand. I just wanted to be sure. And if I got it correctly, I mean, one of the further arguments, I would say, to go for the therapeutic vaccination as compared to the adoptive transfer is that if I saw clearly the slide, in the adoptive transfer, the level of HBe/Ig was remaining stable in some experiments. Is it true?
Mm-hmm. Yeah, that's always something where you have to be careful if you see mouse data, because a drop in S antigen in our hands mostly reflects that antibodies are produced. That means you have anti-S, which neutralizes S. That does not necessarily mean that the infected hepatocytes are eliminated from the liver. So if E antigen does not change, that means there is still a high number of infected hepatocytes in the liver. So there is no really proof that there is a curative effect.
And that was the case, if I understood correctly, with the adoptive transfer.
In the patients, the patients normally are E antigen negative from the beginning.
No, in the animal model.
In the animal model, we lost both E and A.
Okay, so I didn't. Okay,
both was
sorry.
lost. Mm-hmm.
Okay. So we have some Q&A. Maybe you can read them, Ulrike.
Yes. Anders, hi, Anders asked, how about liver immune tolerance and PD-1 inhibitors together with the vaccine? I think that's a very interesting approach. And we actually tested that, again, in mouse models where we applied antibodies against PD-ligand-1 and an siRNA against PD-ligand-1. And interestingly, it was more efficient to use the siRNA than it was to use the antibody, which I found a very interesting result, but definitely combining checkpoint inhibition with therapeutic vaccination is a very interesting approach. And then there's a question by Alan Schmalion. Are overall data consistent with the possibility that antibodies other than classically neutralizing can also have a therapeutic effect against infected cells? Of course, isotype matters and will be influenced by CD4 T cells. That's also a very good question, which I cannot answer, I must say. Definitely, there's the possibility that the antibodies have induce an ADCC. So an antibody-dependent cytotoxic effect in that case. It's very hard for us to test because we can use FC mutated antibodies for therapeutic approaches, but if they are induced in vivo in the mice, there is no way to influence whether those are FC activating or FC non-activating antibodies. But I'm quite convinced that that also plays a role.
I think that those were the questions we had. Do we have other questions? Let's see. Other question. I think just to conclude that your results on hepatocellular carcinoma are obviously very stimulating. The patients you are enrolling presently with for the TERVAC studies, but I guess that for the first studies you are really addressing patients with limited liver lesions. I mean, because you are still concerned by the intensity of the flares and the risk of, but then you can move to patients with more with cirrhosis and.
Yeah, I mean, the patients which the trials are done by SCG cell therapy, not by ourselves. And the patients they target at the moment usually have liver cirrhosis or often have liver cirrhosis, but they always check that they have still a very good liver function. So child A basically stage of liver cirrhosis. Otherwise, we do not include them because apparently the risk would be too high if we target infected hepatocytes. And the lesions in the liver, they are not limited because those are patients who usually have failed first and second line therapy for HCC. So they rather have larger lesions and several lesions. And that's basically as you start off with, if you develop something which is more the last opportunity remaining for those patients. Okay. For the therapeutic vaccination, where we go into chronic carriers, there we of course check that there is no pre-existing ALT or signs for pre-existing inflammatory liver disease, not to undergo any risk here.
Yeah. Okay, well, thank you so much, Ulrike. Great presentations. And it's nice to have a presentation on hepatitis B in the context of the global virus network. So thank you so much. And thank you everybody for attending the meeting. Bye-bye. Thank you.
Thank you very much.